Hello. My name is Denise Montel, president of GSA. We're going to be presenting 3 awards today, starting with one from last year, and that is Dan Hartle of Harvard University, the recipient of the 2019 Thomas Hunt Morgan Medal. The Morgan Medal is awarded to GSA members for lifetime achievement in the field of genetics, recognizing the full body of work of exceptional geneticists. Dan's extraordinarily broad research program combines mathematical models with cutting-edge experimental techniques to tackle important questions in evolutionary biology and genomics. Of course, this very breadth makes it difficult to summarize his accomplishments in just a few minutes. In the words of Colin Meiklejohn, one of the nominators, Dan has employed equations, microbes, fruit flies, and the malaria parasite to discover how genes are organized into genomes, vary within populations, and change over evolutionary time. By combining theory and experiments, he's made seminal contributions to transmission, population, evolutionary, and medical genetics. His early work on segregation, distortion, and Drosophila, followed by studies of experimental evolution. Using chemostats to grow E. coli under controlled conditions, he showed context-dependent fitness effects of naturally occurring protein variants. In analyzing the system, Hartle worked with mathematician Stanley Sawyer to develop a statistical framework for estimating the distribution of selective effects from polymorphism and species divergence data. Their theorem is frequently used in comparative genomics today to analyze species differences in whole genome sequences. His lab's investigations of the effects of transposable elements on genome structure and evolution included the discovery of the widespread Mariner transposon and its use as a tool for transformation. He also played important roles in mapping and sequencing the Drosophila genome. When the lab investigated the evolution of gene expression differences, they found evidence that sexual selection and sexual differentiation are important in driving the evolution of gene expression networks. He has also applied his evolutionary research to the important problem of drug resistance in malaria, investigating the population genetics and evolutionary history of parasite populations. He's also a dedicated teacher, supportive mentor, and the author of more than 500 articles and 30 textbooks. We are short on time and long on accomplishments, so I'll stop there, but I'll hope you'll join me in honoring Dan for his many achievements.
Thank you very much. This is a wonderful honor, and I'm very glad to be able to join this meeting to thank the membership of the GSA. This award will always remind me of the 100 or so graduate students and postdocs and collaborators who actually did the deep thinking and the hard work that this award recognizes. I wish I could thank each of them individually, but I can't. However, you can, there's a complete list that you can find in Genetics, volume 212, on page 362. And it is on behalf of this amazing group of people that I am very proud to accept the honor of the Thomas Hunt Morgan Medal. And I thank the GSA again, and that's all I have to say.
Moving on to some 2020 prizes. First up is Welcome Bender of Harvard Medical School, the 2020 recipient of the Edward Nowitzki Prize. Great. The Nowitzki Prize recognizes a single experimental accomplishment or a body of work in which an exceptional level of creativity and intellectual ingenuity has been used to design and execute scientific experiments to solve a difficult problem in genetics. Welcome Bender exemplifies the spirit of this award through his creativity in revealing the molecular nature and regulation of the bithorax gene complex. This work began when he developed a positional cloning approach in pursuit of interesting Drosophila genes. These included the bithorax complex, in which Ed Lewis famously showed that mutations affecting specific larval body segments were arranged along the genetic map in the same order as the parts of the body they affected. To clone the genes responsible for such intriguing phenotypes, Bender's idea was to gain a foothold by starting with a clone that, although possibly far from the mutation of interest, was at least on the same chromosome. Then they could use it to fish out a larger genomic region overlapping this original clone and use the most distal sequence on that large piece to repeat the process, which came to be known as walking down the chromosome to find the target. This approach allowed him and his colleagues to identify the bithorax complex, and the method was then adopted by geneticists studying other species. It became instrumental in identifying a number of critical pathways and human disease genes. After the long genomic walk to the bithorax complex, Bender and his team went on to characterize its structure and function. Their investigations reconciled Ed Lewis's genetic observations with their molecular biology, finding that the complex only encodes 3 proteins, all homeodomain transcription factors, and the complex is in fact largely made up of complex regulatory domains controlling expression of those few proteins in time and space. They proposed that the sequential activation of the segment-specific functions results from sequential opening of chromatin domains along the chromosome as you move along the body axis. Over his career, Bender and his team have developed many novel and elegant approaches to mapping the bithorax regulatory domains and examining how polycomb repression restricts access to silenced areas of the complex. His ingenious work reflects the great rewards of taking a focused approach to a complex piece of biology. Please join me in congratulating him. Welcome.
Thank you, Denise. I must say I feel unworthy of this award because I know so many of you have made much greater contributions to science and education and the community. But be that as it may, I've been particularly lucky to have had the support and guidance early in my career from Art Chovnik and Ed Lewis, 2 of the greats in fly genetics. Lucky to have been joined by such a talented group of students and postdocs and research assistants. Lucky to have had generous collaborators. Lucky to have joined the Drosophila community with a culture of sharing, and now more than a century of observation and insight. I also should thank the National Institutes of Health for many years of support. Finally, I'd like to share my recipe for satisfaction in science. Whatever field of biology you investigate, you should try to spend some quality time every day with your chosen organism. It helps to renew your sense of wonder. Thanks again.
Finally, Seth Bordenstein of Vanderbilt University is the 2020 recipient of the Elizabeth W. Jones Award for Excellence in Education. The Jones Award recognizes individuals or groups who have had significant sustained impact on genetics education at any level, from K-12 through graduate school and beyond. Bordenstein is honored for the development of Discover the Microbes Within, the Wolbachia Project, an initiative designed to bring real-world scientific research into middle, high school, and college biology classes. Bordenstein's own research focuses on symbiosis, evolution, and host microbiome interactions, often using Wolbachia, which are widespread bacterial endosymbionts of invertebrates. He's a passionate advocate of bringing authentic research to the classroom and has done this through an integrative lab series that engages students in producing new scientific data on Wolbachia. In the words of one of the nominators, Joe Handelsman, copious research now supports his approach as one of the most effective ways to attract and retain students in STEM fields and affect real and lasting learning. In the project, students identify arthropod species in their local fauna. They obtain DNA sequences of potentially new strains of Wolbachia and determine the evolutionary relatedness of these strains to other sequences in the genetic databases. In the beginning, Seth and his colleagues focused on providing training and resources to local educators. But word spread fast, and the project soon expanded to become national and eventually international. Initial funding from HHMI allowed the team to purchase thermocyclers that they could loan to schools, particularly in low resource communities, and provide pipettes and expensive molecular biology reagents. The project is still going strong 15 years since it began. The resources have been translated into other languages. The materials are open source and freely available online, so anyone around the world can use them. Tens of thousands of students, perhaps more, have used the labs in some way. Seth has rightly noted that the project is a team effort and emphasizes the role of his wife, Sarah Bordenstein, who serves as project director of the worldwide program. Leveraging her experience in microbial ecology and high school science education, She has ensured that the work meets curriculum standards, refined the project's online components, and manages day-to-day operations. To both Seth and Sarah, thank you for your dedication to bringing research into the classroom. Seth.
Good morning, everyone. It's an honor to be a part of this event this morning and this community. I think we all know that GSA is a world-class society and community, and I want to thank you and the nominators for recognizing education and outreach efforts taken by so many of us really over the years. Last year, a high school student said, "Science is so much more than what you see in a textbook and on TV." Now that type of student self-awareness never ceases to inspire because that student clearly gets it. They get what we're trying to do. I've been involved in science for 25 years and had an early passion, and that is that generally society vastly underestimates the capacity for kids, even ourselves often, and the public to do great science. My first grant as a new PI was to develop and run this Wolbachia Project Lab Series in middle schools, high schools, and colleges, locally and then nationally and internationally, and especially to underserved schools in greatest need. I couldn't have imagined the diverse ways this first grant changed my perspectives and those of so many others. I want to thank the many who have been crucial to the Wolbachia Project because project-based learning is not effective alone. It has to be embedded in a culture that's powerful. And so thank you to the GSA again for being a powerful recognizer of science education beyond the campus. I want to thank Wolbachia and the animal kingdom for my education and career. And I want to thank colleagues George Wolf, John Warren, Bob Minkley, and Michael Clark, who we all joined together about 15 years ago to found this project. And then I want to thank the teachers, scientists, and students across the world who've been involved with it. And my partner in science and life, Sarah Bordenstein, who is the project director of the program and took the initial idea, made it professional and rewarding for teachers and students. The lab series is all open access and open-hearted online. Thank you again.
Terrific. Well, congratulations to all of you.